And with that, I'd like to actually welcome our final speaker. So Dr. Jens Volkmann is a full professor and chairman at the Department of Neurology at the University Hospital Würzburg in Germany. And again, it seems fitting that after we've talked about etiology, we've talked about research, we've talked about treatment, we've talked about deep brain stimulation, and obviously psychosocial support and some of the triggers as well of dystonia, that we actually conclude the meeting with a look to the future, I think, and a wrap up of the research and the future in terms of the etiology and treatment of dystonia. So with that, Jens, I'll hand over to you.
Yeah, thank you, Rachel. It's a pleasure to be with you. What I'm going to do is I will speak a little bit about a European funding that we had over the last 5 years or so. It started actually briefly before COVID. And this was a consortium that was called EuroDiscover. And there are a number of interesting outcomes of this consortium, which I would like to present today. So I'm going to share my screen. And hope that you all
can see it.
Perfect, perfect. So we have already talked a little bit about the causes of dystonia. So we can mainly categorize the causes in 3 categories. One is there are some patients that are suffering from a so-called monogenetic course of dystonia, which means they harbor a gene which is almost entirely responsible for their dystonia. And one of the examples, for example, is DYT1, and this is a patient that we've treated many years ago, where you see the effects of a DYT1 mutation, which is actually causing severe generalized dystonia. Now, what is important to know is that even if you have that gene, it is by no means sure that you're going to develop dystonia in life, because the penetrance of these genes is typically very low. In the case of DYT1 dystonia, for example, it is only 30%. So only 30% of those that are affected by the gene mutation are in the end getting the dystonia. There is a second group of causes and etiologies which are related to trauma and overuse. So this has been reported for a number of cases with cervical dystonia, that cervical dystonia occurred after injuries, whiplash injuries in car accidents, for example, that after a while the affected individuals develop cervical dystonia. It's also well known for writer's cramp, that writer's cramp is resulting from overuse or musician exercise-induced dystonias, where the repetitive strain injuries and the pain associated with exercise or overuse can actually trigger dystonia. Here, the risk of developing dystonia is very, very low, because there are many people that have these kind of injuries, traumas, and only a small fraction of them is in the end developing a dystonia. So here we have the question of whether there is another factor that can actually explain, in addition to the injury, for example, a genetic risk predisposition, then the evolution and development of dystonia. And finally, brain lesions to brain circuits can actually cause dystonia, either by pharmacological interventions. Certain drugs that act on the dopaminergic system can cause dystonia, or stroke lesions or other lesions inside the brain can actually lead to a brain network dysfunction. And again, this is causing dystonic movements, such as in this patient here, where a traumatic injury, an accident, a car accident, with a bleeding to the basal ganglia actually caused this severe dystonia on the right side. Now, we tried to put all these different causes together in an overarching model, trying to explain dystonia not as a disease, because it is not a disease. It's actually it's a group of diseases that are causing dystonia, but the final common end is a certain syndrome, which means characteristic symptoms that cause the twisting of the body, the abnormal muscle contractions and the postures that are seen in dystonia, and this is irrespective of the body part affected. So the pattern of muscle overactivation is very similar for a patient with writer's cramp or a patient with cervical dystonia or in generalized dystonia. It is just affecting different body parts. And the idea is that there are genetic causes that lead to a certain predisposition, so-called endophenotypes, which may create a certain large-scale network state or molecular or cellular changes, or also changes on the electrical activity of neurons that then predispose in a second step to be sensitive to environmental factors that can trigger the onset of dystonia. So basically, if you harbor the gene, if you harbor the DYT1 mutation, and you have all these endophenotypes, the predispositions to develop dystonia, you may either end up having basically a normal function and being asymptomatic because you didn't have an additional environmental factor, a certain stressor, or your brain cannot compensate anymore because it is basically brought out of balance by a certain environmental factor, a certain trigger, and then this leads to the development of dystonia. So I already said that there are a number of factors that have already been identified, such as overuse or trauma, which can then lead to this kind of decompensation. And the main objective of the consortium, the EuroDiscover consortium that I'm speaking about, was exactly to evaluate this hypothesis of the development of dystonia. So we wanted to explore the disease mechanisms of dystonia and identify common and novel pathophysiological pathways on a molecular level, a cellular and microcircuitry level, and on the level of large-scale basal ganglia cortical networks, on whole brain networks. And we wanted to explore the pathophysiology and disease progression in different types of dystonia, so-called monogenic forms, genetic forms of dystonia, but also sporadic forms of dystonia, to identify novel clinical biomarkers by using various techniques such as fMRI or non-invasive magnetoencephalography recordings, or invasive techniques such as deep brain stimulation. And all this was supposed to be supported by an animal model, by a rodent model of dystonia, which should basically mimic and help us to explain many of the pathways and sequences that we are not able to investigate in humans. So this is the consortium that I'm speaking about. It was a consortium of different labs across Europe. I'm starting with Albino Oliveira Maia from Portugal. He is a neurophysiologist that was working on mouse basal ganglia electrophysiology in this consortium. Antonio Pisani, he was also working on the mouse electrophysiology on so-called slice recordings. Andrea Kühn from Charité in Germany was contributing clinical studies and deep brain stimulation studies. Robert Jech from the Czech Republic in Prague University Hospital was doing clinical studies and functional MRI studies on patients with deep brain stimulation electrodes to see which networks are affected by deep brain stimulation. Marie Vidayer from Paris was contributing again clinical studies in genetic dystonia. And we had a contribution of Dystonia Europe to dissipate the results and bring the results through the patient organization to the patients, which was headed by Monica Benson, the executive director of Dystonia Europe. And you see myself coordinating these projects, and in particular contributing a novel rat and mouse model, which was used in this case. So the idea basically of our novel model approach is when you have this kind of gene, such as DYT1, you actually need an interaction between this gene and some kind of unknown environmental factor to bring up dystonia. And this is something we try to study in a mouse model. So we had access to mice that are actually having exactly the mutation that you find in humans. So they were genetically engineered to have the TOR1A mutation, the DYT1 mutation, that that is the cause of generalized dystonia in humans. And as you see here, these mice are absolutely normal. So they don't show any dystonia. But we had the hypothesis that you need actually a second hit, a second trigger, an environmental trigger to make them dystonic. And this was evaluated in initial experiments. We created either a nerve injury to the hind limb, and this caused, when the animals were recovering from the injury, this caused actually dystonic movements of the hind limb. And this dystonia was progressing and was then spreading to the other leg, demonstrating that you could indeed trigger dystonia by an environmental trigger. The other way of triggering this dystonia is by letting the animals perform a similar movement thousands of times per day. This is similar to musicians' dystonia, let's say, where you have to press, this is, of course, you cannot let the mice play piano. So here they just have to press a lever and they get a little reward every time that they press the lever. And this incentivizes them to press the lever many, many thousand times per day. And this is, again, causing deterioration of their finger movements, and they become dystonic, the movements. And again, this can be shown very nicely in recordings. Now here we had the model. So we basically have animals with a genetic predisposition, which can be compared to animals that do not have this genetic predisposition, normal animals. And we have an environmental trigger. And now we can compare basically 4 groups: animals that have the genetic predisposition compared to animals that do not have the genetic predisposition, and then the impact of the environmental trigger on animals with and without the DYT1 gene. And again, here is the idea basically that you have to distinguish between the disease mechanisms and the symptom mechanisms of dystonia. So in the beginning, we basically have genetic abnormalities and environmental factors, and they lead to common changes on the molecular level, the microcircuitry level, which means electrical activity level, and then the level of large-scale brain networks, which become abnormal and produce abnormal activity, which is then output to the periphery and is causing dystonic movements and dystonic postures. So we are talking here about disease mechanisms, but also the mechanisms of how dystonia evolves. And even if we may not be able to provide directly a cure for dystonia, a huge victory would already be if we can basically understand how the brain compensates in those individuals that harbor the gene but do not get dystonia, and how eventually by therapy we can actually influence the large-scale networks so that the dystonia symptoms go away or are not even expressed to start with. And for this, we are using a large number of different techniques in these animals where we can compare the genetic predisposition and finally the onset of dystonia and the network changes. We can do transcriptomics, which means we can look at gene and protein expression. We can look at morphology, how the brain changes through the impact of the environmental trigger and with the onset of dystonia. We can measure neurotransmitters. We can look at brain function using PET, for example, glucose PET, and look at metabolism. And we can try interventions by cell type specific neuromodulation. So these are very modern techniques where we can basically suppress or activate certain cell types by so-called optogenetics or pharmacogenetics. And all this was part of the Dystonia Europe Consortium. Yeah, some first results to, as basically a look into the lab. So our colleagues in Porto, in Portugal, they used very fancy, elaborate methods to look at the activity of particular cell types in the striatum in a region that is involved in the basal ganglia, in motor learning and plasticity. And they were able to demonstrate that certain cells during the movements in the animals show a differential activity in animals that do not have a genetic predisposition, as opposed to animals that have the genetic predisposition but do not exhibit dystonia. And then when they exhibit dystonia, suddenly the cell activity is markedly changing. So this is a change in cellular activity which is purely associated with the expression of dystonia, with the upcoming of dystonia, but not with the genetic predisposition for dystonia. And this indicates that there is a common pathway, basically, which then leads to the symptom expression, regardless of whether you have a gene or no gene that is causing dystonia. Again, our colleagues in Italy have been able to show that these changes on the cellular level in the striatum can lead to abnormal plasticity. And abnormal plasticity is one of the causes that we presume, and many have many divergent evidences already that abnormal plasticity in the basal ganglia is one of the causes of the network dysfunction associated with dystonia. We can even use deep brain stimulation to treat these animals, and we can evaluate the impact of the treatment on the movements. These are the finger movements made during the lever press in dystonia animals, and show that basically deep brain stimulation can normalize this function. And this also allows us now to probe new areas in the brain for their effectiveness for deep brain stimulation, apart from the well-known globus pallidus or the subthalamic nucleus, to find out whether there are probably more optimal nodes to suppress dystonia, or even cause some longer lasting changes that are not reversible when you switch off the stimulation. And finally, our colleagues in Prague undertook very interesting efforts to compare the network activity using fMRI in patients with and without pallidal stimulation, to better understand which areas need to be activated or deactivated during successful deep brain stimulation. Now, one of the inherent problems that we have in dystonia research is that there is no good biomarker for what is dystonia. Dystonia is usually rated by clinicians. If a doctor would tell you, well, this is dystonia, and this is such and such severe, and this is not another type of movement disorder. But for scientific research, this is typically not enough. For scientific research, you need to quantify the movements, and you need to be able to track them over time. And we had to prove, when we wanted to publish our results, that it was not only looking like dystonia, because many causes can be mistaken or basically cause cramping or a malpositioning of the fingers or hand or the paw of an animal. For example, pain could lead to an abnormal positioning of the hand. And the authors, basically the reviewers of the paper, urged us to find a method to quantify and objectively evaluate dystonia. And for this, we developed, we took a method which was already available at that time. It's a video kinematic tool where we can actually use the video recordings and place markers on video recordings to evaluate the movement trajectories in 3D space. And we could demonstrate that the movement trajectories of animals that were performing the movements normally and those that were having dystonia were very different. And the difference was in a physical measure that is called entropy. Entropy is a measure which demonstrates the orderless of a movement. So it basically tells you whether a state is very ordered or very disordered. And the degree of disorderness is something you can measure in physical terms by a variable which is called entropy. And this entropy differed greatly between movements of normal animals and dystonic animals. So this is the pipeline here that was used. It's called Deep Lab Cut. It's a video kinematic analysis, which basically helps you to identify all kinds of movements in rodents, in rats, and it's used by many labs across the world. But here we specifically trained this algorithm, which is an artificial intelligence algorithm, on videos of paw movements of dystonic animals and got very clear results. Now, this kind of application has also been used now for rating of cervical dystonia videos. And this is an example here. We use basically videos from our database of a study that we performed some time ago on the effectiveness of deep brain stimulation in cervical dystonia. These videos, together with the twister scorings that we have for these videos, were analyzed for the movement state, for head angle, and we were basically capable of automatically reproducing with this video analysis the twister score in an objective manner. But we were also able to find some factors that you can normally not find in clinical scales that distinguish patients that have a good outcome after surgery from those patients that will have a less favorable outcome after surgery. And these measures were typically measures of the dystonic mobility. So we found that subjects, individuals that had a lot of mobility before surgery, a lot of head movement before surgery, were actually having better outcomes after surgery than those that had predominantly a fixed posture. Not saying that patients with fixed postures did not respond at all, but the outcomes were more favorable of those which had dystonic movements. And this was recently published. And we are now looking into the possibility of basically using this method to create a kind of an app or software which will allow physicians to objectively rate dystonia and use maybe these kind of features to guide their botulinum toxin treatment or to guide deep brain stimulation therapy. So in summary, we know that dystonia is not a single disease, but it is a group of diseases that cause, in the end, a common network dysfunction, which is the cause of dystonic postures and dystonic movements. And we have now a new rodent dystonia model available, which allows us to study the interaction between genetic causes of dystonia and environmental causes, and potentially also to find molecular interventions very early in the disease path, which help to prevent or strengthen compensation and prevent the start of abnormal plasticity and then the onset of dystonia. And as the side effect of the consortium, we found a new analysis method which allows objective measurement of dystonia and can even be used in the clinic, for example, for cervical dystonia rating. So this was my lecture, and I thank you very much, and I'm open for questions.
Hello, that was brilliant. Thank you. We do have a couple of questions before we close. And thank you for coming in right on time as well. And the question, I'm going to wrap up a couple of questions in one, if that makes sense. Is there always a genetic predisposition coupled with an environmental or physical trigger? Do you think it's always both, or do you think that it can be one or another that triggers dystonia?
Yeah, I think there is an interaction. But very clearly, I mean, there are some that have a very strong genetic cause, which are genes that cause dystonia and maybe a weak environmental trigger. And in other patients, it will be a very weak genetic contribution that is not found in a single gene, but maybe in a group of different genes that make you more susceptible to certain environmental triggers, and then a very strong environmental trigger. So it is an interaction of both, I assume. But the contribution, the individual contribution, can be on different scales.
Brilliant. And final question from Simple. Again, I'm going to wrap up a couple of questions into one. I mean, how can people find out if they have this genetic predisposition, other than perhaps understanding that members of their family had dystonia? Is there any gene typing that can be done?
Yeah, the familial, the family history is not very reliable in dystonia because, as I said, it can express in some generations and it may jump other generations, and still the gene is present because the expression is so low. Even in DYT1, which is probably the strongest genetic cause, only 30% of those that have the gene get affected. So it can simply skip generations and be dormant, so to speak. So the only way to find out if you have a genetic predisposition is to do genetic testing. And there are panels available that can test multiple genes. There are now more than 20 known dystonia genes, and they're all screened. And this gives you an answer whether you have one of these different genes. There is no immediate treatment effect, though. This is why in some countries it may not be paid by the health insurance. We did genetic testing within a consortium in Germany where we paid for it out of research funds.
Okay, brilliant. And actually, a really interesting one. I said the last one, but this is really interesting. Are you aware of any interesting advances in non-invasive brain stimulation?
Yeah, this consortium led to another consortium, another funding that we obtained, which is called Retune. Retune is a large consortium in Germany that is funded with almost 50 million euro, where we study neuromodulation in general, invasive and non-invasive neuromodulation. And there is one of the projects deals with a new form of non-invasive neurostimulation, where basically electrical surface stimulation is targeted in a way that different beams of surface stimulation lead to deep nuclei stimulation inside the brain. And the idea is that we can modulate abnormal plasticity, which is leading to dystonia. Whether this is going to have an immediate clinical benefit, we don't know. So far it is only experimental, but we will have the results in the next 4 years.
Perfect. Thank you. And again, thank you again to Jens, Dr. Volkmann, for rounding off, I think, 3 hours, 3.5 hours of presentations. And it's only right that you actually close it with a look to the future and a look to the ongoing research. And as I said to the previous speaker, actually in 2 years' time, we'll probably see presentations on this, but taking it further. So I look forward to seeing those as well in the next couple of years to see where the research has landed. So thank you so much for concluding the
My
meeting.
pleasure.
Thank
Thank
you.
you.